Hi, my name's Amber. I'm a PhD student from the University of Leeds in the United Kingdom, and I'd like to share with you a project I'm working on supervised by Professor David Westhead. My poster presents a statistical approach integrating multi-omics data, which aims to characterise the cis-regulatory interactions governing plasma cell differentiation in humans. The framework we developed here combines community detection with penalized regression to predict how cis-regulatory elements come together to control transcription of genes driving plasma cell fate. Cis-regulatory elements are short genomic sequences which control gene expression. Enhancers, for example, upregulate expression of their target gene through the binding of activator transcription factors. Gene regulation is a coordinated effort between multiple coacting cis-regulatory elements, the gene promoter, and a variety of transcription factors. Cis-regulatory elements can be quite tricky to find. There are estimated to be hundreds of thousands of them in the human genome, mostly in non-coding regions distal to their gene. However, computational approaches, fueled by next-generation sequencing data, can predict these cis-regulatory elements from their biological features. One such feature is chromatin accessibility. Cis-regulatory elements are cleared of nucleosomes to make way for transcription factor binding. We can measure accessibility of chromatin using a technique called ATAC-seq, which sequences regions of DNA that are depleted of nucleosomes. With ATAC-seq, we can also detect transcription factor binding by looking for something called transcription factor footprints. These footprints are dips in read coverage where cleavage of the TN5 transposase, the enzyme used in ATAC-seq, is blocked by transcription factor occupancy. ATAC-seq and RNA-seq experiments have been performed on B cells, which have been stimulated to undergo plasma cell differentiation in vitro. From this study, we have a time course dataset with samples taken from 3 donors across 9 time points. Both epigenomic and transcriptomic datasets are input to our workflow to predict communities of coacting cis-regulatory elements that are important to differentiation. This novel approach of community detection and penalized regression was first developed here in our group and has shown success applied to hematopoietic datasets. Applying this method to our plasma cell dataset, we first identify candidate cis-regulatory elements from the ATAC-seq data. These are regions of DNA which are differentially accessible across the time course and within 100 kilobases of a gene's transcription start site. These represent potential cis-regulatory elements whose activity patterns shift as the cell differentiates. We next characterise the chromatin accessibility and transcription factor binding profiles of each candidate and use InfoMap community detection to find communities of similar candidate elements. Finally, we input the accessibility profiles of these communities and lone candidates alongside gene expression patterns from the RNA-seq data into gene-specific LASSO linear regression models. Here we model the relationship between chromatin accessibility and gene expression, where gene expression is the response variable Y and chromatin accessibilities for each candidate are the predictor variables X. LASSO is a form of penalized regression, which is able to shrink the coefficients of predictor variables down to 0. By doing this, LASSO performs something called variable selection, resulting in a sparse model where only the candidates most predictive of gene expression remain. The candidate elements selected by each model that are found to be statistically significant are predicted to regulate that gene. Now we can use these models to understand how genes are regulated, but first we need to be confident in the ability of this method to predict true cis-regulatory elements. One way we can validate our predictions in silico is by looking at mutation data. When cis-regulatory elements become mutated, gene expression can be perturbed, leading to altered protein production, abnormal cellular processes, and ultimately diseases like cancer. And as a result, GWAS studies often find quite a lot of malignancy-linked mutations in non-coding DNA, which localizes to cis-regulatory elements. So we created over 1,000 models for differentially accessible transcription factors, those whose dynamic activity drives differentiation. And we looked at the COSMIC database for non-coding somatic mutations in cancer. Here we find that predicted candidates are significantly enriched in mutations linked to B cell and plasma cell neoplasms when compared to the candidates we reject. So this result shows promise, and we're continuing to analyse mutation data from other sources for further validation. Now, to give an example of what kind of regions we are predicting, here are the results for a gene that has been described as the master regulator of plasma cell differentiation, BLIMP1. For BLIMP1, we have predicted a community of 3 enhancers, all downstream of the gene promoter, with transcription factor footprints that indicate the community could be co-bound by OCT and RUNX transcription factors in the later stages of differentiation, which are the cell types when the enhancers are most active and BLIMP1 expression is greatest. And this here exemplifies our method's focus on this cooperational mode of gene regulation, which is often overlooked by other enhancer prediction methods. As part of our validation strategy, we now plan to analyse high C chromosome conformation data to find out whether these community members are in contact with each other and their gene promoter. We look forward to thoroughly validating this work with the high C data, experimentally validated enhancers, and cis-regulatory mutations, so that they can then go and confidently use these models to yield insight into cis-regulatory control of cellular differentiation. Thank you for listening, and I'd like to finish off with giving thanks to all these people for making this work possible.